All right. Hello, everybody. Good evening. Nice to see all of you here on a busy February Thursday, the last day of February. A happy leap year day to all of you. Welcome to UCR Palm Desert Center. My name is Agam Patel, Executive Director here. I'm so happy to see all of you here and welcome you to our three-part lecture series with our friends at UCR's Division of Biomedical Sciences. We're so happy to have Dr. Monica Carson back here in the desert. For those of you that might remember, pre-pandemic, we did a women's lecture series here with Dr. Carson. We're happy to be working again with her office. Before we bring her up on the stage, please allow me to share some important upcoming events. The next two lectures in this series, in the biomedical lecture series, will be on March 26th with none other than David Lowe and Keziah Israel. And then on April 2nd, we'll have Dr. Carson herself and Dr. Zeng. So please mark your calendars for those two in this lecture series. Flyers are outside, and we'd love to see you here then. On March 5th, next week, we are introducing our new science lecture series with our friends at the College of Natural and Agricultural Sciences. We're going to be talking with Dr. Robert Allen, leading climatology professor, and he is going to be talking about the ever-changing climate and how to avert the crisis. On March 6th, we are talking with our friends at the UCR's Jewish lecture series, Jewish Studies. We're talking about the Holocaust of Biafra, anti-Semitism, and genocide in sub-Saharan Africa. This is part of our ongoing collaboration with UCR's Jewish lecture series. So mark your calendar for that. We're still talking about next week, folks, so hang in there. March 7th, we're going to be joined by Joshua Tree National Park Rangers, David Larson and Stacy Manson, who'll be talking about night skies and light pollution. So we're going to be joined by some National Park Rangers here. So a little bit of everything here for those of you that are interested. On March 14th, as part of our Boyd Deep Canyon lecture series, we're going to be joined by Dr. Doug Kelt of the Department of Wildlife, Fish and Conservation, who is going to be talking about the longest running experiment in the Southern Hemisphere. So that should be an interesting talk. My thanks to the UCR Palm Desert Center partners. Raise your hands, please. I know I've seen a bunch of you here. Thank you. Thank you for supporting UCR Palm Desert. None of what we do out here would be possible without your support. So we thank you from the bottom of our heart. If you'd like to learn a little bit more about the Palm Desert Center Partner Program, please come and see me or Laura Williamson. All right. Allow me to introduce Dr. Carson. Dr. Monica J. Carson is the S. Sue Johnson Presidential Endowed Chair in the glial neuronal interactions and serves as the professor and chair of the Division of Biomedical Sciences at the University of California, Riverside School of Medicine. She additionally serves as the director of the Center for the Glial Interactions and is the editor-in-chief for the Journal of Neuroinflammation. She has been an active member of both the American International Neurochemistry Societies since graduate school, serving in multiple roles, including president of each society. She received her AB degree from Bryn Mawr College and her PhD from the University of Pennsylvania. Her research focuses on identifying CNS immune system interactions with a specific focus on the role of a microglia in supporting lifelong brain health and how genetics, inflammation, and environmental insults trigger and or exacerbate neurodegenerative disorders. Dr. Carson's choice of research focus was strongly influenced by her early interactions as a student with the National Multiple Sclerosis Society and by individuals affected by MS. Please join me in welcoming Dr. Carson.
Thank you so much for that really kind introduction. And what's embarrassing, that introduction is longer than what I'm going to say up here, which is good because we're going to get to the meat of what's going on. I just wanted to thank all of you for coming here and really welcoming us back, since it was pre-COVID when we were able, as the Division of Biomedical Science, the School of Medicine, do our health and wellness series and start connecting with all of you again. And so we're really delighted to come out, and we are featuring in this series of 3 lectures some of our centers. And our centers in the School of Medicine are all about bridging the science to the health. And that's what you're going to hear in the talks that we're doing. And so you're going to hear 2 brief talks today, and I'm not even going to say anything about them because they're going to come right up here. But in the future ones, we're going to be doing things about air quality, how actually the air that you're breathing right now is affecting you, and on the health of your whole system and some other things that are going on. And then in the final session, it'll be a session about brain health, basically from birth to death. Hopefully it's a good journey, and what we can do to optimize it. But today, when we do this and we come to the questions, we're really hoping you'll ask all the questions you have, curiosities about what they're actually presenting, other questions that are in that area that you're interested, or how do our centers work? Those are all fair game. And the other thing we'd like to know, and hopefully we'll have some surveys later, is what are you interested as the community in having us do research in? What questions should we be asking? And so we want to begin that long conversation. So without further ado, I would like to introduce the person who's going to moderate all of these and introduce our 2 speakers today, Professor Scott Pagan. He's a very outstanding researcher, but he doesn't get to tell about his research today. But he is our vice chair of the Division of Biomedical Sciences. He oversees our research and centers, keeping us all compliant and, getting us all enthusiastic about coming together. So he's going to come up right now. Let's just welcome him, because I'm not going to go long on this, but welcome him to carry the day.
Thanks, Monica, for that lovely intro. So we don't want to prolong the meet too much. So I have the pleasure of introducing our first speaker, and then we'll have a short, brief talk with our second speaker, and then we'll have them come over here for answering questions session. So I have the pleasure, of course, introducing Professor Nick DiPatrizio, who is the founding director of our Center on Cannabinoid Research. This is one of our youngest centers. He's been working diligently hard over the last couple of years and has made a tremendous impact, and he's going to share a little bit about what the center's about, as well as also probably highlight his upcoming symposium, where he's already attracted not only leaders in California in the cannabinoid field, but also nationwide. So without further ado, I'm going to introduce Nick DiPatrizio so he can tell us all the good, great things he's been working on in his center in cannabinoids. Nick?
Thank you, Dr. Pagan. Okay, thank you for the introduction, Dr. Pagan. So as Dr. Pagan mentioned, today we're going to talk a bit about cannabis. And in particular, what is our body's natural cannabis? Okay? And we'll take a tour of cannabinoid research at UCR. Now, let's see here. Okay. So before we get going, I wanted to highlight, as Dr. Pagan mentioned, UCR School of Medicine's newest center called the UCRCCR. Just rolls off the tongue very nicely, doesn't it? The UCR Center for Cannabinoid Research. So very exciting time for cannabinoid research. So the mission of our new center is we're comprised of scientists and clinicians from across campus and with common interests in advancing our understanding of roles for the endocannabinoid system, so this natural system in our body, how it plays a role in health as well as disease. And then, of course, we look at the impact of how cannabis impacts these different pathways that sort of hijack this system in the body. But before we move on to some of the fun science, as Dr. Pagan mentioned, we're having our inaugural symposium focused on cannabinoid science and community health, bridging the gap. So we have leaders from almost all the centers of cannabinoid research at all the UC campuses and medical schools coming. This is going to be on Friday, May 3rd. So keep an eye out for this. Here's our webpage for the Cannabinoid Center. We have all of our information on here for the upcoming symposium as well. Everyone here, of course, will be invited to come. This is open to the public. Okay? So let's get into it. So my laboratory has several different fun projects in different areas of research emphasis, but they're all really centered around identifying roles for the endocannabinoid system in health and disease. One major project in the lab is looking at how the gut and the brain talk to each other and, for example, drive us to overeat tasty foods, and how the cannabinoid system plays a role in this normal physiology, and in fact in the pathology also of metabolic syndrome, and how these systems become dysregulated. We also look at, with Dr. Zlebnik's lab, another assistant professor in our division, we look at how the endocannabinoid system controls food intake as well as reward. So what makes those foods taste good, taste bad, and we'll touch at the end on just a slide or two showing some future work identifying roles for central reward mechanisms in mediating the drive to eat from the gut. We also look at whole body energy metabolism. We look at gut barrier function. So, for example, in inflammatory bowel diseases, the gut barrier can become compromised. And we look at roles for the endocannabinoid system in these processes. We also look at, with Dr. Nayer's lab, host-pathogen interactions, so interactions between us and pathogens and bugs out there, and look for roles, again, for the endocannabinoid system in these processes. But importantly, we look at the impact of cannabis across all these processes. So for today's talk, we're just going to focus on these first two here, gut-brain signaling, as well as food intake and reward, and roles for the endocannabinoid system in these processes. So what is the endocannabinoid system? Okay, you can see in this word, it almost looks like cannabis, right? So endo is endogenous, so from within. So this is our natural endogenous cannabinoid system from within our body. Cannabis hijacks this system. Think about different chemicals in cannabis as the keys floating around, and the locks are these receptors on the surface of cells. So we produce natural cannabinoids that are the keys that fit into these locks. But cannabis also has chemicals that hijack this system, different keys, but they access the same lock. And in some ways, in certain circumstances, can impact the system similarly to endocannabinoids or differentially to how endocannabinoids work. So this system in the body's really gained recent attention for key roles in the seeking, sensing, as well as consumption of natural reinforcers such as palatable foods, so tasty, fatty, kind of sugary foods that we all love. And the system, of course, may become dysregulated in diet-induced obesity, and we'll go through some of the data, some recent data from the laboratory highlighting these pathways. So the endocannabinoid system in both the brain and in central nervous system, as well as in our peripheral organs, in fact, control food intake, energy homeostasis, and reward. As neuroscientists, we a lot of times can become so brain-centric and neurocentric and forget that there's a body attached to our head. And in fact, our behavior all day long is being influenced by signals coming from the periphery. And this can be without awareness. And we're going to touch on some of these signals as we move through. So for this series of studies, we investigated roles for gut-brain endocannabinoid signaling in food intake and reward, and its dysregulation in diet-induced obesity. So, of course, there will not be any quizzes after this on these pathways. I just want to highlight that there are metabolic pathways in the body for the biosynthesis and degradation of those natural keys. One of them is called 2-AG and one's called anandamide. But notice what happens. These are the keys. These activate the same locks that the intoxicating ingredient in cannabis can activate. So in this way, cannabis is hijacking the natural reward system, the natural cannabinoid system. I just wanted to show some brief data real quick to sort of set up the endocannabinoid system. So we've all heard of the munchies from cannabis, this pretty reproducible effect from cannabis, right? The right dosing can induce overeating and consumption of, in particular, foods that taste really, really well. So this was some early work of mine when I was an undergraduate student at Temple University, where we would give rats THC and then look at, very simply, the intake of a fat diet. And I'm not going to delve into some very interesting things that we found, but nonetheless, for the first couple days with this dose, they get the munchies. Then over time, in fact, the system can adapt, and we have a whole big giant project looking at what goes on there. Well, what I wanted to highlight is how these receptors work. THC activates this receptor. How do we know this? Okay? So we run pharmacological experiments. We give the key that activates this cannabinoid receptor, and if we want to say, is THC working through the cannabinoid 1 receptor, for example? We pre-treat the mice then, in this case, these were rats, with a key, but you break it off in the lock. So it's not active anymore. Now what happens? The THC can't activate that lock anymore. And look what happens. You give a selective key that breaks off in the CB1 receptor, and look what happened. We completely blocked the effects of THC, which tells you that there's a specific receptor mechanism involved. But one other sort of point here I want to highlight is when you give that clogger alone, it shuts off feeding. No THC around, but when you break off a key in that lock and don't turn it on, but just break it off so nothing can bind it, you shut off feeding. This tells you that the body's producing natural chemicals that are binding that lock normally and driving the food intake. Okay, so this is a bit of a segue into some of the questions that we'll touch on here. So the first is, we'll probably only get through the first 2 or 3 of these questions here, but nonetheless, the first is there a role for gut-brain endocannabinoid signaling in diet-induced obesity? How do endocannabinoids in the gut control this signaling? How does the brain control endocannabinoid production in the gut? It's this bidirectional pathway. It's a two-way highway communication between the brain and the gut. And we look at each of those pathways. And then at the end, if we have time, how do endocannabinoids in the gut control preferences for these high-fat, high-sugar diets? So the first set of experiments I'm going to go through here was spearheaded by, in fact, my first PhD student in my laboratory, Donovan Argetta. He's out there now killing it, just about to get a faculty position and has won all these wonderful awards and grants, and I'm very proud of him. Nonetheless, he asked the question, what happens to the endocannabinoid system in the body in diet-induced obesity? So here we use a mouse model, okay, to be able to identify mechanism of how things work in the body. We use rodent models. We feed them a very tasty, high-fat, high-sugar diet. This mimics the Western diet. And no big surprise here, they gain rapid body weight, okay? So these are the mice on that diet, gain rapid body weight. Again, no big surprise. Why are they gaining more body weight? They're eating more, okay? So we next asked, what happens to the endocannabinoid system? We know this system in the gut, for example. I won't go give all the 10 years of background on that prior to these studies, but nonetheless, we've identified in the gut, this system can drive overeating. So we asked the question, is this system hyperactive in the gut lining? And does that possibly drive that overeating that we see in diet-induced obesity? This is using these fancy techniques in the laboratory of mass spectrometry. And we're basically able to, all you need to look at is this number is much higher than this number. Okay? In those obese mice, in the lining of the gut, okay, this signal, these natural cannabinoids, this is an endocannabinoid. This is that 2-AG. This is that body's natural cannabis. Spikes. Okay, so these diet-induced obesity is associated with increased levels of an endocannabinoid, which happens to be a monoacyl glycerol, in the mouse upper small intestinal epithelium, and this would suggest possible dysregulation of endocannabinoid metabolism. Good, I took those slides out. We also worked out a lot of the metabolic pathways, which we won't dive into here. So that was some fun biochemistry, but what does this mean behaviorally? Did that signal in the gut actually drive overeating? So to test that question, in this series of studies, we used this peripherally restricted cannabinoid clogger. So remember, we were saying you could give THC, activate the system, or you can give a key that break it off in the lock and it blocks the system. This is a blocker. So remember, the cannabinoid levels naturally are elevated in the gut. We give a chemical that breaks off in that lock and stops that signaling from happening. These are mice that are obese, okay? So these are the normal, we call it a vehicle treatment. This would be the control. So basically just water giving them, right? And they consume this many calories over 8 hours, how many hours are we looking here? 6 hours. We give that drug that blocks that effect, blocks that elevation in cannabinoids. And look what happens. We significantly reduce food intake of these obese mice. But what I want to highlight is in mice that are lean, the system isn't active. The drug does nothing. There's no endogenous keys floating around. There's nothing. You break it off in there and it doesn't matter. But look what we did to the obese mice. We reversed their phenotype to that of a lean mouse. Okay? So we reversed overeating by blocking the system in the gut. So overeating associated with diet-induced obesity is driven by increases in peripheral endocannabinoid signaling. So you may be asking, how does this signal get from the gut to the brain? Right? How does the gut talk to the brain? So there's several different highway systems that can be used. One, just blood, right? Organs can release chemicals, these endocrine signals that can communicate with the brain. However, there's this nerve called the vagus nerve. A very interesting nerve. This nerve talks from the gut, tells the brain what's going on in the gut, and then there's another nerve from the brain going back down to the gut to tell the gut what's going on in the brain. So we focused on that system to ask, is endocannabinoids manipulating the vagus nerve? So one more bit of information. This whole system, this is a cell in the gut. When you eat food, it's sensed by cells lining the intestine. It's sensed. And it sends a signal through to the vagus nerve to tell you to stop eating, right? Why do we keep eating? Why don't we just keep eating when something tastes so good? Eventually we get full, and that sensation of fullness is driven in part by release of certain chemicals. And I'm not going to go through just for the sake of time here, but I'm going to tell you, I'll give the whole punchline away of this series of work. What we showed is the elevated endocannabinoids in the gut block satiation. So in the obese mice, they overeat because they're not feeling full. The cannabinoid receptors block the communication from the nutrient sensing to the vagus nerve and to the brain. So normally we eat food. Again, the gut senses it, triggers to the brain to stop eating. That whole process is completely dysregulated in obesity. And we have a whole lot of evidence for that. So since we have about 5 minutes left, I'm going to skip this is a big giant paper we have very hopefully coming out in Journal of Neuroscience soon, so I'm not going to go through that. Nonetheless, we identified some of the pathways from the brain to the gut, these cholinergic pathways that become overactive in diet-induced obesity. Okay, let me just end on one more series of studies here. So we want to ask, does this signal actually control preference? So if we give mice, or you, let's say, access to just some crummy, fiber-rich food or access to a cheeseburger, you're going to prefer the cheeseburger, okay? Or whatever you prefer, fats, sugars. So we do the same thing in mice, and this was spearheaded again by Donovan in the lab and a recent graduate of the lab as well, Brian Avalos. So we gave the mice this choice between burgers and lettuce, okay? But it's a little more, you don't actually give them lettuce. They're little pellets that mimic all this stuff. The mice love this stuff. They just absolutely love it. You saw how quickly they gain body weight, right? So what we did is we gave a clogger of the cannabinoid system. This clogger floats through the body. It accesses the brain. It accesses the guts also. And what we do is, here is the preference for that diet. So you can see the bar, if at 50%, if you were at 50%, that means you eat the same amount of this as the same amount of that. You're indifferent to them, right? Normal mice and us with vehicle, with the control, are going to significantly and greatly prefer this over this. When we block the cannabinoid system, look, we block the preference. Now that was giving a drug. We developed a mouse model where they're missing the lock. The keys can float around, but the lock is missing only in the intestinal lining. Those mice, when you give them a preference test, it's completely blocked the preference. So this is telling you that endocannabinoid receptors, those locks in the intestinal lining, are required for acute preferences for Western diet in mice. Now, just looking ahead in the last final minute here, we have a new fun collaboration with Dr. Zlebnik in the laboratories I mentioned in the introduction, a new assistant professor, wonderful neuroscientist in our division. So we wanted to ask the question of what is going on in the brain? Is this signal in the gut controlling dopamine? We've all heard of dopamine, right? So dopamine's sort of associated with that craving and that wanting and forming behaviors to obtain natural or non-natural reinforcers. This is that system in drug abuse that gets highly dysregulated. So the craving becomes amplified. So during certain behaviors that are rewarding, you're going to have dopamine release in these reward areas in the brain. And we use this fun grab dopamine sensor in mice where you're actually able to record dopamine being released in that area of the brain that controls reward. So again, we started with pharmacology here, and we said, okay, using a peripheral cannabinoid receptor antagonist, so clogging the receptor, breaking off the key in it. This one was developed by a colleague of mine, Alex Macrianis. Nonetheless, this drug doesn't cross into the brain, but it also blocks preference, as you can see here. And look what happens to dopamine. This is control condition. This is when the mice are approaching that bowl to go eat the preferred diet. Normal mice with no drug on board, cannabinoids are being produced. They have this dopamine spike that is maintained. But so that's these mice here that are preferring the diet. When we give that cannabinoid clogger in the peripheral clogger, look, you get this brief spike and then the dopamine response is gone. Okay? Suggesting that the cannabinoid system in the gut is controlling reward and central reward processes. So the system in the gut's upregulated in diet-induced obesity, which drives overeating. Endocannabinoids in the gut communicate with the brain and promote overeating and obesity by this mechanism that includes inhibiting nutrient-induced satiation. So remember, cannabinoids blocking you from feeling full, blocking satiation feeling. Overactive production is controlled by this cholinergic pathway that we've just delineated, and these receptors in the intestinal epithelium are required for Western diet-induced preferences in mice. So the system, as you could imagine, may be a good target for safe anti-obesity drugs that do not directly interact with the brain, and we can chat about this later. And these drugs may be then devoid of the psychiatric side effects that we can talk about that have been found with drugs that are brain permeant and can disrupt directly cognition. So we believe we can remotely control the brain and behavior from the gut. And of course, I just have to thank everybody, current and past folks from my laboratory, and please go over and visit our center, cannabinoid.ucr.edu, and thank you to all the funding. All right, thank you very much.
You just leave that on there, we'll just turn it back.
Okay. Yeah. You gotta have that on for the. Okay, thanks, Dr. Patricio, for giving us that intro into cannabinoids and all the different flavors associated with it. So hopefully you'll be able to pick that up in the question and answer session afterwards. We're going to now have the pleasure of introducing our next speaker, and he's a man of many hats. I have to actually look at the list because there's so many on this list. But, of course, he serves as Associate Dean for Pre-Clerkship in our medical education, as well as our Director of the Pharmacology thread. But we were able to steal him away tonight for his other role, which is the Founding Director of the Molecular MOMED, Molecular Translational Medicine Center, and also his, make sure I pronounce it correctly, President of our Meta Labs is one of his startups that has spawned from the research that he's going to be able to share with us today. He's also the Daniel Hayes Endowed Chair for Cancer Research, and in that role, he's also been a major player in bringing a new National Institute of Health Cancer Center funded grant to UCR in collaboration with City of Hope, and I think he's going to share many of those different insights with us tonight. So without further ado, I'll introduce Maurizio Pellecchia, our distinguished professor.
Thank you, Scott, and thank you. I know it's getting dinner time, and after Dr. DiPatrizio, it's a pretty tough act to follow. Dr. DiPatrizio makes me already want to eat some more. So I need to talk about something a little bit more serious, though, unfortunately, that is cancer. And as you know, there are many hallmarks of cancers, and I want to point out that academic research is really at the forefront of the new therapeutics that we see today. And I want to make a few points about how academic research has really made an impact. Now, How do cancer cells become cancerous? We don't actually have an answer. I wish we had a silver bullet and we had the perfect target. It's really a very complex alteration of molecular circuitries that really leads cancer cells to sustain proliferation, resisting to cell death. Some solid tumors, they require vasculature, so angiogenesis is very important. Some, they become immortal, and there are other effectors that they suppress and make them resisting to chemotherapy. So most chemotherapy really works on trying to slow down proliferation of cancer cells, simply so. And so how can you slow down proliferation? You try to slow down cell division. And then anything that cells need to divide is mostly synthesizing DNA. So that's the most of the anti-cancer agents, they really tend to target DNA synthesis with anti-metabolites or molecules that they prevent the biosynthesis of DNA. Basically, they starve cancer cells from DNA biosynthesis, and therefore they cannot replicate. Other chemotherapeutic agents, they target cell divisions, for example, antimitotic agents such as Taxol or paclitaxel. I'll talk about that in a second. So what's the problem with chemotherapy? Well, they are not specific. So they work. We all need to take them if we have cancer because they seem to work, especially in slowing down proliferation initially. But again, it's not cancer specific. So they cause severe side effects, and those toxicity are more immediate on those tissues that they tend to proliferate naturally faster. The hair follicles, in my case, the GI tract as well, the mucosas, and as well as the bone marrow. So, red blood cells, platelet, and white blood cells, they replicate very, very rapidly. So they get affected by chemotherapy, in addition to other specific, agent-specific toxicities. So now, where do academic research comes into play? Well, all those hallmarks of cancers, they do come from misregulation of signal transductions. So all those are different genes that they are tightly regulated in a normal cell, so that the cells has a lifespan. It will grow, a tissue, an organ, for example, tissue homeostasis. We have a certain size of our organs. They don't become much, much bigger because all those are tightly regulated. So what happens that in cancer cells, sometimes there is some misregulation. Some of those oncogenes, they can get highly expressed or can get activated. And for example, they can push in the accelerator and signal this cell that has to proliferate. So for example, a few years ago, in Philadelphia was discovered that in a type of leukemia, you have a chromosomal translocation that generates this oncoprotein, BCR-ABL, along this proliferative pathway. And so studying the exact structure of this oncogene, this protein that is present only in leukemic cells now, using structural studies that again come from academia, including the three-dimensional structure of how this BCR-ABL gene looks like, scientists then at Novartis were able to design a lock, a key actually, that goes into this lock and jams it, so that this oncogene no longer can work. It can no longer signal proliferation. It was 1998, and this was the first targeted therapeutic, because now it's targeting an oncogene that is present only in those leukemic cells. Since then, we have kept studying many of those circuitry. In particular, the field has been on proliferation circuitry. We are more interested in motility circuitry because what makes cancer cells want to detach from the primary tumor, extravasate, get into circulation, and adhere to other tissues will really usually kill the patients. This process is called metastasis. So, but since then, was 1998, now patients, when they're lucky, they have, we can identify some specific genes that they are main driver on their particular cancer. For those, there is a number of targeted therapeutics, at least several dozens. I don't know if I could click this one here. Is this connected to the internet? Oh, yes, I can. Skip verification. Oh, but I don't see it here. So this will connect you with the NCI page where you will see dozens of targeted therapeutics for different cancer types. So cancer patients now, they can benefit from genetic testing to see if their cancer can be reconduced. That is not always the case, unfortunately, to a specific gene or a specific group of genes for which we already have a targeted therapeutic. So this is really going down to personalized medicine. So academic research plays a role in all stages of, I see something is happening here. Okay, here. So at UCR, we really try to marry all the technology and the basic sciences. The basic sciences will allow us to identify possible new oncogenes, new targets, and the technologies really can help us develop new therapeutic modalities, not necessarily having a small molecule drug, but also other type of modalities of inhibiting those oncogenes. So the center was put together a few years ago, founded in 2016, is really aimed to do a number of activities around research in translational medicine, not only in oncology, also in other areas, but oncology is becoming our expertise in particular. So some of the activities, when we started, this was supported by the School of Medicine, our dean, and then later by the Office of the President grant of about $2 million, where funneled most of these funds into the MRB1 incubator space that is housing potential UCR startup companies in translational medicine. We go into that in a little bit. We supported many proof of concept grants to UCR faculty. We bring companies to see our research so they can sponsor some of the studies that we do at UCR. We do annual symposia. We recently received a large NCI-sponsored grant to partner our studies with the City of Hope, and I go into that in a second. And again, the intent of the center is to support and catalyze new ideas, collaboration, funding initiatives in translational molecular medicine, in particular in oncology. So again, most of the academic research resides on these two areas where you have basic sciences, where you have cell biology to identify new oncogenes and new targets in different disease areas, but in oncology in particular, we marry that with molecular technologies that they are ways on how to attack those oncogenes, how to devise new therapeutics. But then it's not the end of the story, because we still want to develop those into potential clinical candidates. We need to go into this painful process, which is drug development, that usually requires the intervention of a biotech company or a pharmaceutical company. And then some of those could be our own companies, our own spin-off from UCR. That's the idea. So I want to give you an example. I mentioned we are targeting metastases. So what we really look at the circuitries that are involved in tumor metastases, we realize that one of the receptor tyrosine kinases, called EphA2, is really critical in promoting cell migration and cell proliferation. I'll show you a few examples later. So in a normal situation, the EphA2 receptor tyrosine kinase is engaged in the heparin ligands from another cell. So as a cell-cell contact is important for cell adhesion and to keep tissue homeostasis. Now, what happens is in cancer cells, in particular in pancreatic, prostate, breast, and lung that we have been studying, EphA2 becomes overexpressed. So there is an excess of EphA2 receptor versus the ligand, and this really signaling cell migration and metastasis. Because those cancer cells, if they have this receptor unbound to the ligand, they are looking for a ligand. So basically that excess of the receptor provides the cancer cells this property of moving, of migrating, and also to adhere to other tissues. So, in our lab, we developed a molecule which we call targefrin, because it's targeting the heparin receptor, that really not only binds to the EphA2, it causes the dimerization of the receptor, and the receptor gets internalized and degraded. So we can really restore the level of EphA2 in a cancer cell to the level of a normal cell, of a healthy cell. So this was a journey that took about 12 years to get to this point where we get this molecule. And really, let's see if I can make this one to work. What really was enabling is the technology of looking at things at the atomic level using NMR spectroscopy, X-ray crystallography, and these type of techniques and biophysical approaches. They really gave us the ability to fine-tune the molecular structure of targefrin to the point that is very, very effective in different models. I'm not going to describe those in detail, but I have a couple of examples. So our indication for now is pancreatic cancer, because pancreatic cancer really depends on EphA2. So the majority of patients, they have elevated EphA2. Unfortunately, they will die within 1, maximum 2 years from diagnosis. A few patients, they tend to have lower EphA2 because they are lucky, and they tend to live much, much longer. So our idea is that we can restore the EphA2 level by suppressing EphA2 level with targefrin. We can bring this cohort of patients from this very rapid mortality to a much higher survival rate. So this is how we measure cell migration. So now basically those are pancreatic cancer cells. You see them. You can create a space between them in a well, and then with a microscope you can see what happens after time. And then you can untreat the cells, and then you can treat them with targefrin. And let's see if this one starts. So you see this is the time that is passing. It's a time lapse. We take a picture like every hour with a little movie. So you see that the aggressive pancreatic cancer cells, they tend to migrate. They really are unhappy because they have all this EphA2 and they want to find the ephrin ligand. And they try to find it by just invading any space they find. When you treat them with targefrin, therefore the EphA2 receptor now is suppressed and is silenced, you see that they are sluggish, they don't want to go anywhere, they just lump on each other, on one another. So this is a very simple assay that we can measure as a surrogate of how the compound would be predicted to be anti-metastatic because you're blocking the cell migration. So I'm doing more technical data. So if you do that, of course, with all the statistics, etc., you can check. You can do a different concentration. That basically the molecule behaves pretty much as a surrogate ephrin ligand, which is what you want to do. So, and again, as I mentioned earlier, when the receptor is activated by the molecule, this causes degradation of the EphA2 receptor, which we can read in a normal experiment like this. So we are doing two things. We are not only blocking the receptor, but also causing its own degradation, which is basically eliminating this oncogene from the surface of pancreatic cancer cells. So does this translate in a mouse? So when we treat mice now with pancreatic, with the orthotopic models of pancreatic cancer, and then we can measure liver metastasis, we see that the mice that they receive the molecule, they have much reduced, almost no liver metastasis. I also mentioned that this effect is very important for prostate cancer as well. Prostate cancer cells, usually EphA2 is actually used to take prostate cancer cells and make them aggressive by transfecting EphA2 gene artificially in it to study them in laboratory metastasis. So, in fact, what we also notice that the recurrence of EphA2 of prostate cancer is also correlated with the EphA2 expression. So patients that they have elevated EphA2, they have a much higher probability that within 10 years they will rehab recurrence of prostate cancer. So same experiment that we did, we treated mice in orthotopic model of prostate cancer, and we can see clearly that when they are treated with our targefrin, we have the same effect as the entire ephrin ligand in suppressing tumor metastasis. Now, the other thing that came to mind is this. So when this is the receptor, and is bound to targefrin, the entire receptor gets internalized, goes to a lysosomal pathway, and gets degraded. So that means we can use this to actually deliver a payload selectively, like some of the antibody drug conjugates that they are available now. So what we decided to do then, we can conjugate our targefrin with a cytotoxic agent such as Taxol, as I mentioned earlier, an anti-mitotic, a chemotherapeutic agent. And then this will deliver the Taxol directly to cancer cells, any solid tumors that they express EphA2, without delivering that to normal cells that they don't have EphA2 expressed, or if it's expressed, is engaged with the ephrin ligand. So really, targefrin has at least 3 different targeting modalities. The one using targefrin itself to suppress the level of EphA2, which should block cell migration and should block metastasis, can be used in addition to standard of care or between cycle of standard of care. Or we can develop targefrin chemotherapy conjugates because the targefrin will activate the receptor and bring the entire chemotherapy selectively to cancer cells. So when we probe this hypothesis now on breast cancer, for example, model, this is a genetic model of triple negative breast cancer, you see that develop very massive lung lesions, lung metastasis, but when they are treated with targefrin, targeted Taxol with targefrin, we nearly see no lung metastasis. And most interestingly here, it's in collaboration with the Cedars-Sinai, we also measure circulating tumor cells. They have a method to measure circulating tumor cells from blood samples, and we can see that the number of circulating tumor cells in patients that are treated with standard of care is much higher compared with the targefrin conjugated with the chemotherapy, because again, those circulating cells, they depend on EphA2, so they can be captured by this agent, and then the payload, which is a cytotoxic agent, will kill them. More recently, we did the same experiment with pancreatic cancer models, and again, we had, in this case, we conjugated with the ephrin dimer again, targefrin, and also with the monometic one that does not cause a degradation, but we can have a cleavable linker there that will concentrate the payload in the proximity of the pancreatic cancer. And again, the take-home message here is that we can have a lower concentration of Taxol in the conjugate because, again, it's accumulating on the tumor side through the PHA2 receptor and butyrylglycan. So, as I mentioned earlier, one of the activities that MolMed was involved in was to support the creation of the MRB1 incubator space that was inaugurated in 2021. So the idea was to support and incentivize advancing research from UCR into the clinic, right? So can we start biotech companies that they can take now those discoveries that they are still at the laboratory stage, preclinical stage, and translate them all the way to the clinic? And why not? Doing that, supporting also the economic development of Riverside County. Surprisingly, UCR is a university of this magnitude. We still have very little in terms of biotech companies in our county. So this is what we are trying to catalyze that with this initiative. So I had to walk the walk, and I started Armida Lab a couple of years ago. So Armida Labs is really the idea behind Armida Labs is to take the patents that we have at UCR, fold them into a spin-off company. We have laboratory space into the incubator space, and then we are trying really to develop those all the way from preclinical studies to potential new drug applications and to clinical trials. So this is the team. We have a brand new CEO, Paul Mola, that just joined us a couple of months ago. And we actually submitted a National Cancer Institute SBIR grant, so we got the first award. The first phase 1 was awarded about a couple of months ago, and we have a second one on the way. So we really put together the entire infrastructure on how to develop targefrin. This is actually the molecule made by a company in San Diego. They actually manufacture targefrin that can be used for our IND-enabling studies. And currently the company is at the still seed stage. We are looking for VC funds and angel investors to develop these all the way to the clinic. Now, I'm almost over. Just one slide. So I mentioned earlier, so we just received this National Cancer Institute grant. So this is really an idea that we had to partner with the City of Hope and to try to bring all the expertise and infrastructure that the City of Hope, that has a comprehensive cancer center, including doing clinical trials in oncology, all the way to marry them with our discoveries and research at UCR, in particular with an emphasis to help increase diversity in cancer research, both from the point of view of the scientists that are involved in cancer research and also to address health disparity in cancer's outcomes. So this was just awarded in August, I believe. August 2023, we got about $13 million grants. And again, those are the three major aims: translational research, identification in oncology research, keeps changing on its own. And then drug discovery and development between UCR and City of Hope, that they have abilities to do IND-enabling studies and CRO facilities, and then clinical trials, the City of Hope mostly, but perhaps also in Riverside in oncology. And these are all the folks that they helped in this project, and I'd be happy to take any questions when we go over there. Yeah.
Thanks, Maurizio. Why don't you take a seat? Wonderful timing. Nick, you can join us up here. Sorry, a little slow up the stairs there, a little old Army injury I'm getting over. So, but really great to have these two guys. And Maurizio, of course, is being a little humble there. He's not only spearheaded the incubator space, but the SBIR that he received, I think, was one of the first SBIRs that University of California, Riverside had actually garnered in the advancement and development of this therapeutic. So now we'll take questions from pretty much any range you want. You've seen some of the science. You know, these guys are been through it about how to bring a drug, to where it's at, how to bring a center, and how to bring public awareness in these areas of cancers and cannabinoids. Who's as we'd say in the Army, lean forward in the foxhole and ask the first question. There we go.
I was just wondering, how long do you think it's going to be before, when you have a patient who's developed a cancer, that you can do a genetic analysis of that particular individual and create a regimen of drug treatment based on your development for that specific individual, unique to that person?
Right. So part of that, is that too loud? Part of that is already happening. So when patients are diagnosed with cancer, my recommendation is always to take, it's called a Tempus. There are other companies that they do that. Many of the advanced clinics, they know that. They can take your biopsy, send it for genetic analysis. And there is at least about 40, 50, 60 genes that if they are altered, so there is mutations in the EGFR, mutation on ALK. I put a link there. You can see all of them. So immediately they will tell the oncologist what type of treatments are available for the patient, what type of regimens have been recommended based on previous trials for the patient. So that is already in place. The problem is that not all the genes, not all the oncogenes have a drug for, and not all the cancers, they are characterized only by one or two genes. Okay, so that's the major problem. So the NCI a few years ago did something that is, I believe, almost finished, which is called the NCI match. So rather than treating patients with going cancer by cancer, so normally you have an indication and you have a set of drugs for that particular indication. You do first standard of care, and then standard of care at certain point doesn't work anymore, and you try something else. So they try really to match the genetics of the cancer with the drugs, rather than going cancer by cancer by cancer. So that's kind of going where you want to go. Now, the ideal, which we are not there yet, the real answer to your question is, can I take a biopsy, study it, and see if a particular treatment would be more beneficial for a patient than another? It makes a lot of sense to me to do that, but the surrogate assays that we have in the laboratory, they're not directly translatable to an outcome in a patient, okay? Because what we do in a Petri dish is eliminates a lot of variables. I give you an example, pancreatic cancer, right? Pancreatic cancer, we can work with cell lines, everything seems to work perfectly. However, the pancreas is surrounded by these fibrous tissues, and a lot of cancer-associated fibroblasts that they express a lot of proteins, a lot of extracellular matrix. So it's very, very complex. So your drug may work very well in a Petri dish, but it may never reach the tissue. It may never penetrate it. So that's why the field is moving from cancer cells to, so the way drugs were tested in oncology has been like this. I test them in a cancer cell, in a naked cell, and then you see if it works. Okay, most of the time they seem to work. The next level, though, is to create a 3D spheroid, a 3D, instead of having cells in monolayers, is a 3D mass, that perhaps more recapitulated tumor. That's a little bit closer to a human, but still not enough. Then we can generate little organoids, a mini pancreas. That's a little bit closer, but still not enough. So what the field is really going is transgenic mice. So for example, we work with a mouse model, which is called the KPC, where the mouse is really mutated in a gene called the KRAS. And within about 6 to 8 months, the mouse will develop pancreatic cancer. So now that's a model that is much, much closer. Now, how do you go from that to a human? Well, it's possible. So you could take human biopsies, you can culture them and implant them in a mouse, and then study the human biopsy in a mouse. Okay. This is all great. Is it quick enough? Is it effective enough? And are the statistics enough that you could do it patient by patient by patient? The answer today, unfortunately, is no. Okay. So the first answer I gave you is probably the most accurate. Do a genetic analysis of the biopsy, see if there are genes for which there are already drugs or clinical trials on the drug, and that's the best avenue. All the other I described with those models are still too slow and not practical to be doing on a patient basis. Sorry for the long answer.
Thanks, Maurizio. Anybody else? Oh, we got one over there.
I can do it. Hi. It's really for both of you because you're doing wonderful research, fighting obesity and, curing cancer, but there must be other schools and everybody's doing research. Do you guys talk to each other, or is everybody in their own little bubble doing their own thing?
Yeah, I think we both want to start.
Yeah, so, both of our laboratories are very highly collaborative, so I love these sort of collaborations. We're experts in certain areas, but we can't do it all. So we collaborate with folks that are experts in another area. For example, I gave an example of measuring dopamine in the brain. I collaborated with a colleague on campus here for that. Across the UC system, one of the major drivers for our new center is to be the hub. So this is sort of the build out here, the dream, to be the hub for cannabinoid research throughout the whole University of California system. Okay. So in doing that, we hope to foster collaborations a lot more with cannabinoid research. But most of my work is highly collaborative with folks from really all over the world.
Yeah.
Yep.
Yeah. Can I ask another question really quick? It sounded like I always say the word, right? Cannabinoid is sort of not a good thing in your gut, where I thought it was like the master regulator that brought your body to homeostasis. I thought all that was good in your body.
And all of that's right.
Oh.
Yeah, I like to think of the endocannabinoid system as nature's safety net. It's there to, for example, if you're leading to a hyperexcitable state, lots of glutamates and excitatory neurotransmitter in the brain leading up to a seizure, our cells can produce endocannabinoids to slow down that neurotransmission. So that's one example where it's a benefit. It's also a benefit to overeat because it's feast or famine. We haven't evolved out of this yet. So you always imagine ourselves kind of running around the savannah. And if we taste something that tastes good, we want to eat it. Well, it's this system that then becomes dysregulated in metabolic syndrome and can lead to overeating and obesity. So it's sort of this natural protective pathway is for survival acutely, but over time, if it becomes dysregulated and we have McDonald's down the street, access to these high levels of calories at any time throughout our day, it may be disadvantageous, right? So it's advantageous for a survival from an evolutionary perspective to have the system selected by evolution, right? But now in modern societies where we have just quick access, it can become hyperactive and in fact has been targeted therapeutics. I could just give you a quick anecdote if you don't mind. So you may be thinking, did we just cure obesity here, right? And some of these molecules, in fact, are going to be going through clinical trials. The last one I showed you is going for liver fibrosis now. But nonetheless, there was a drug on the market in Europe. It was called Acomplia. And it was a clogger of that receptor. Remember, activate the receptor, munchies, eat, block the system, stop eating, and reduce body weight and food intake. So it was out on the market. The chemical was a rimonabant. And in the States, it was about 2 years behind from getting full FDA approval. This drug worked wonders, reduced waist circumference, improvements in a whole bunch of different metabolic parameters, worked wonders. The problem is when thousands of people started taking this drug, they realized there were increased psychiatric episodes, increased rates of depression, and several extra suicides in the group that got the drug. So the FDA, of course, caught wind of this and they nixed the whole entire program. Sanofi Aventis was just ready to recuperate some of their money from investing billions in this molecule. So that is a segue to what we had talked about here, where this work is attractive and these molecules are attractive if you tweak them so they don't access the brain and lead to these psychiatric side effects. The cannabinoid system in the brain is regulating mood and emotion and all these other behavioral states. So we don't want to mess directly with that. We want to remotely control it from the gut and perhaps then have the same beneficial therapeutic and metabolic effects, but without those psychiatric side effects that are inherent to brain permeant molecules. So yeah, thank you for the question.
Yeah, and I'd like to go back to also the first question about the sharing, and that's really one of the things that our centers are really about, is sharing not only with you guys, but also sharing as nexuses for sharing within the research themselves. Like Nick's center here, he showed you, of course, really some great research, but there's also other facets of his center, such as public outreach, public health, and I think how many members are you up to now?
I think we're 15, 16 members now. Yeah, composed of physicians and so clinicians as well as scientists from across campus. So really exciting time.
So yeah, so he's kind of his center's really taking it all the way from the molecular level to the impacts on society for some of these treatments and therapeutics. So it was really great answer. And then also Maurizio's, his center does the same thing for us. He, as he mentioned, he's been a great nexus for UCR, bringing a lot of pharmaceutical companies to UCR to kind of exchange great ideas with folks like Nick and a lot of other faculty in translating these, but maybe he can mention a little bit about that.
Right. We also collaborate with all the other UC. So UCR is part of the UC-wide Drug Discovery Consortium. We meet monthly and we discuss issues about new targets, new way of targeting them, new way of funding opportunities that we can have in translational medicine. And then the most important to me, frankly, is really collaborating with more clinical sites that they are doing clinical trials. We collaborate a lot with the Cedars-Sinai. In Los Angeles, we collaborate now. We have this huge collaboration with the City of Hope, with this very large grant that we just got. So, no, it's a formidable task. Developing a therapeutic or even being part of that is a formidable task. You can't do it alone. It's impossible. No single laboratory. You can make a difference. You can put a piece of the puzzle, like, for example, the Philadelphia chromosome that was discovered at UPenn, right? So that started the entire field of targeted therapeutics. Without the discovery that one particular gene is responsible for CML. But again, those scientists didn't develop Gleevec, didn't develop imatinib, right? So it's really a highly collaborative. Even within the drug discovery process, you have so many different disciplines that they go from biophysics, chemistry, biochemistry, cell biology, cancer biology, animal models, that no single laboratory can do everything. So it has to be highly collaborative. Yeah.
Thanks, Maurizio. Any other questions? Yep, got one back. Oh, right there.
Hi, this might not be your area, but I'm curious about the Gallery blood test and how it can tell you if there's cancer and what kind of cancer is in your body.
Yeah, different cancer types. I mean, of course, for leukemia, blood tests work fairly well, right? Even a simple pathologist can do that. For other type of cancers, markers, they're not really good right now. So we have only a handful of biomarkers that they can give you an indication. EphA2, so our company is trying to develop a biomarker in EphA2 because when pancreatic cancers are very diffuse in the pancreas, they start to shed this receptor. So you can find fragments of EphA2 in the blood. That's why in my slide I had a collaboration with Tulane universities because they developed an assay to develop. But except for a few other markers, unfortunately, biomarkers, in fact, if you go to your clinician, they will not recommend some of them because the error is very, very high. The reproducibility is very low. So the only ones that I can think of, of course, leukemias and lymphomas, they are obvious because that's a pathology that you can see in the microscope. PSA for prostate cancer, that seems to be a very good indicator, but that's it. There are not very many other markers that one can use. So the other area of the field that, for example, Cedars-Sinai they're trying to do, but is not yet an approved method, is to capturing circulating tumor cells using other biomarkers. And so, but again, none of this is FDA approved yet, but is something that may be coming in the future. So having ways to capture circulating tumor cells based on some specific biomarkers that the cells will carry, for example, EphA2. But none of that is approved yet.
Thank you.
All right, next up over there. Yep.
Yes, I'm curious about the collaborative process. A few years ago, someone here, one of the presenters said he wishes there was a way to know what didn't work. It would seem to me, if I understood him, that everybody's very anxious to publish successes, but they don't publish failures. What prevents all this research from going down dead ends and spending a lot of money on things that maybe shouldn't be pursued?
Nick, it looks like you're on the spot.
Yeah, that's good. Really good question here. We can get kind of deep into this one. So without getting too technical, let's say you run an experiment, and the question is, did this experiment work? But what do we mean by did it work? As long as it was run correctly, meaning the person ran the experiment according to protocol, just like a robot, it worked no matter what the outcome is. So a lot of times what winds up happening, and everyone has their own opinions about this, and there's 100 different ways of looking at this, but one is this. What do you do with negative data? Is I think sort of your question. If we test a hypothesis, and the hypothesis is endocannabinoids drive food intake. Okay, we test that hypothesis through a series of experiments. Well, what happens if the result is, no, it doesn't regulate food intake, or it only regulates sugar intake? Okay? That's important data. But what do we do with the negative data? If it didn't work, air quotes, and there's just like one example. If it didn't work, meaning the result's negative, the null hypothesis was correct and the hypothesis is incorrect, right? And that it doesn't drive this food intake. Again, that information is very important. But you really need to contrast it with positive data, is what I'm getting at. So if all of a sudden, all these experiments aren't working, meaning, that it's a negative result, all the hypotheses are wrong, that's valuable information. But as the lead of a lab and scientists, we always want to say, okay, are we sure that we just didn't run the experiment wrong, so to say, right? So we want to come back then and then ask the question from a different angle, recruit another colleague that can come in with a new perspective. I guess that's just sort of one example of what we do. So we published a couple times negative data, but just had to be sure that it's negative, meaning that it didn't support the hypothesis. Somewhat of a long-winded answer. I hope that kind of got a little bit to what you were thinking there. And there's 100 ways of looking at this. So Dr. Pellecchia.
In oncology, you publish a lot of times negative data, unfortunately, right? Because you can't predict, the way I was describing you earlier, the models that we can study. So it's an iterative process. If I could sit on a computer and say, this is the drug, we will be living thousands of years. It's not possible. You make an hypothesis, then you need to experimentally verify. And the experiment will tell you if your hypothesis was correct or not, and sometimes it's gray, maybe partially correct. So you need to go through many iterations of hypothesis, testing, and now your result is as good as your test. So if your test is a clear-cut test, let's say, yes, if I have this assay that will tell me this drug will work in a human, then this iteration, I need to do it only a couple of times and I'm happy. Unfortunately, the test, as I explained earlier briefly, is not going to recapitulate what happens in a human. So what do I need to do is, I do the experiments and I go to the test, the best test I can. The test has to be reproducible and quick and inexpensive. So you can go these iterations many, many times. So then I can go to the next level. Can I test this now in an animal model, for example? And then I find a surprise. It was working in my previous test, but in an animal model it's not working. That's a negative data. But it's not because I wasted time, because I need to study why it's not working. Perhaps the drug is not reaching the proper tissue. Perhaps it's going to the brain where you wanted to go to the gut, right? So, and then you go back to the drawing board. That piece of data is giving you information, say, okay, now I need to engineer out, for example, that the drug is, I don't want it to go to the brain. How do I do that? And so on and so forth. So it may appear, though, that, there's so much failure. We go through a lot of failure. Absolutely. That's most of the time we go. If I could predict 100%, we probably won't be here. We are not needed, right? So most of the time you go through, but the failure will teach you what to do next most of the time. So rather than publishing the failure, you want to treasure that information for the next iterations until you get to that success, right?
Yeah, and you figure out how a system works. I like to tell the students by figuring out how it doesn't work. Right? So if you look in most of our publications, it doesn't work this way. It doesn't work this way, but it works this way. Right? And that becomes more powerful then. If all of a sudden everything started working, that was too easy of a system to figure out, right? We wouldn't even have a job, right? So it's, and then as you mature as a scientist, and I always tell the students, advance through their training, they get a little bit better at testing. We get better at testing the hypothesis as you move along. But still, 9 out of 10 times, the beginning, the hypothesis is incorrect. And that that's science. That's why we do this. We don't know it going into it we don't know the answer from the beginning. And it's this iterative process that is science and what is so valuable, and then the peer-reviewed process, and there's a lot of failed,
and negative data is still valuable as long as it's something that you know why it was negative. Right. And you treasure that information. You use it not to make the same mistake when you do the next iteration, right? So, many anti-cancer agents they're not effective in the end. Because again, we can't predict until you do that clinical trial. You cannot predict what will happen. Our body will try to eliminate the drug as soon as you give it to a patient, right? So, and we cannot predict. We can try. We can have surrogate laboratory experiments. We can have isolated liver microsomes to see how the drug is metabolized. We can give it to an animal, to a laboratory animal, but we have completely different metabolic enzymes between animals and humans, right? We can take human plasma and see what happens to the drug in human plasma. Again, when you go to there's different populations, they have different enzymes. For example, I like the 2C9. I like one of the cytochromes. So if I take any CNS drug, it stays in my body forever. So I cannot take anything. So every person is different in that respect too. So it's very difficult to be able to say, yes, I did all my experiment properly. This is going to be the outcome. No, you would not know. You can only hope that that's the outcome. And then you observe the outcome and you learn from that outcome. If it's positive, fantastic. If it's not, you try to, what do you need to do next to avoid the problem that you encounter? Yeah.
Yeah, I think that's, kind of, but overall on that, that's when you publish something, that's what you're looking for. You're looking, right now the system's incentivizing you to figure out and drive forward and take that negative data and find the direction you need to find to make that next discovery. And when you get to that level, then you've made that next discovery and you can go to the next set of questions. But really great answers, guys. Is there another question, perhaps?
Yes.
Hi, thanks. I've just got two very quick questions. The first one is, for those of us who really like Western diets, how long to your work in endocannabinoids will actually go into a clinical type of phase?
Yeah, good question. So I brought up that one molecule rimonabant, where it was a cannabinoid receptor blocker. It was going through clinical trials, got nixed by the FDA in the United States. So to 100-mile high view here with that in mind, the cannabinoid field has had a bit of a stigma to it over decades. This didn't help anything by these side effects by blocking the system coming out. So a lot of pharmaceutical companies have been hesitant, from what I'm hearing, to move forward with cannabinoid-based manipulating the cannabinoid system for therapeutic value. With that said, this one model, what I can tell you, because I think it's fun, is AM6545 is for liver fibrosis. So it's like advanced stages of clinical trials now for liver fibrosis by blocking that system. With regards to food intake, we'll see. Everyone's trying to come up with their own peripherally restricted CB1 antagonist now. But it just takes time. Once it enters into clinical trials, it's at least 7 years in the minimum before it would come out to the market. So we're at that sort of discovery phase now, discovering the physiology, discovering how it goes from a normal state to a dysregulated state, and then try and revert it back.
So if I understand correctly, if it gets approved, let's just say for another purpose, then you could use it or research it for kind of an off-label type of approach.
Well, I think physicians would have discretion of using it off-label for different pathologies. I like to think of these drugs as tools. We're using that drug as a tool to probe the system and ask, how does it work or how does it not work, right? So we use them as tools, and through that process of saying, this tool works pretty well, well, then someone like amazing Dr. Pellecchia comes along and says, now let's tweak that molecule and make it even have a higher affinity, make it even better, right? So then that's the drug discovery that Dr. Pellecchia can talk a lot more about than I can. So I like to think of our work as sort of, it's at that preclinical translational spot, but we're just making discoveries of how the system works so that the wonderful chemists can come along and make new molecules. But that one molecule I just mentioned, AM, it's Alex Macriana. So we have a big grant together, and he's like the world leader in making all these cannabinoid molecules. So he has so much going on that I probably don't even know about, things that are being patented, and he's got this from here to the end of the hallway of patents for tweaking this system. I just haven't been directly involved in bringing anything to the market yet. I just get so excited about the discovery and all that sort of stuff, and then hopefully Dr. Pellecchia someday we can move something forward.
Thank you.
Yeah, I think Dr. Pellecchia would probably comment. when you talk about 5 to 7 years, it's not like everyone's standing still for those times. There's a lot of steps that these things are going through in terms of toxicology, pharmacology, to get them really dialed in and get them ready for that particular clinical aspect. But Dr. Pellecchia can probably comment on that.
Yeah, so what we do mostly in academic drug discovery is we get to what Dr. DiPatrizio is doing is target identification, right? So like the Philadelphia chromosome that I mentioned earlier. So that started the entire field of targeting. So what the fundamental discovery that was, right? But if you ask those professors at UPenn, okay, when is the drug? And they'd be like, I don't know, right? So there was Novartis. There was a 50,000 scientist company that made the first drug, right? So it's not a simple thing going from a gene, a discovery, but those are the fundamental discoveries that tells the scientist in the pharma company or the biotech or in academia, that's the gene that you want to target. If you were to find a drug against this gene that doesn't go to the brain, etc., and all the things that Dr. DiPatrizio was telling you, then you will have a drug. It may take 10 years to do that. It's not a simple task. Now, when we do also academic drug discovery ourselves, which we could do, and I have many examples, but I mean, I don't know if you heard of the gene KRAS, was in my slide. KRAS is really central to a lot of solid tumors. For many years, industry and pharma companies, they thought it's impossible to find a drug against KRAS. They just gave up. Although it's really central to the majority of solid tumors, until a scientist, the UCSF, in his laboratory, a peer, decided to study KRAS in his laboratory, and he found a prototype molecule and a way to inhibit the KRAS. It's a special activating mutation that has a cysteine instead of a glycine. He published this paper in 2018, Nature Cell Biology, with a prototype molecule. Again, is that a drug? No, it's not a drug. It was a prototype. It's just demonstrating, yes, you can inhibit KRAS. And look what happened to cancer cells. Look what happened in a mouse with the KRAS mutation. Amgen immediately looked at that paper. Within a year, they came up with an optimized molecule, which is called Lumakras. It is doing wonders and has been approved by the FDA 2 years later. So, going back to planet Earth, we are academics, right? So we don't have a company like Amgen or Novartis that can do the work we are doing. So what happens to our projects, and this is a sad way of describing it, but that's what everyone uses it, is that they get to a certain point when you do academic drug discovery, which is called the valley of death of the project, which means you did everything you could possibly do as an academic, right? And either you have a beautiful bridge to a pharmaceutical company to go over all these tox studies that you need to do to be able to start a clinical trial, or the project ends there. Now, I don't see it that way, frankly, because again, okay, it's the death of the project, but all that information that we have published, for example, my colleague Kamal Shokat at UCSF, publishing that prototype KRAS inhibitor, that molecule itself didn't go anywhere, but Amgen took it over, right? So that per se is not really the death of the project. Now, in some cases, though, it may be satisfactory, it may be exciting to go all the way to the clinic, and that's what our center is trying to do, right? So that's why we are trying to network with pharmaceutical companies that they come and see what we are doing so they can catalyze that transition over the valley of death. Or, as a colleague of mine, also UCSF, say, instead of building a beautiful bridge, we can fill up the valley of death with dirt and we can walk across. So that's what we are doing with my small company, for example. We can get small SBIR grants, we have some small donations, etc., trying to get ourselves work with clinical investigators so we can have an investigator-initiated trial, perhaps sponsored by the Sinai or the CDVOP, etc. So that's what we are trying to do.
That was very interesting. Thank you. I just have one very quick question.
Please.
So where are you at in terms of cancer research with using computational methods, and specifically because AI is so topical now, where you can take targeted therapies and look at different phenotypes, I don't know, red hair, or short index finger, like a whole multitude of different types of phenotypes, and not without establishing a causal relationship, but just a coincidental relationship, using the power of these computational techniques and say, oh, this is what we need to do. Where are we at with something, or is that even
perhaps we can get some of those questions afterwards. I think folks things, and so I think I would like to give one round of applause for Dr. Pellecchia and Nick.